everyone. Today I'll be presenting a foundational paper titled An mRNA Structure That Controls Gene Expression by Binding FMN, published by Winkler and colleagues. This study was one of the first to provide clear biochemical evidence that RNA alone, without the help of proteins, can directly sense a small metabolite and then regulate gene expression in response. Specifically, it describes how a conserved RNA element known as the RFN element functions as a riboswitch, which is a structured RNA that binds flavin mononucleotide, or FMN, and then alters the expression of a bacterial genes involved in FMN biosynthesis and transport. Over the next few slides, I'll walk through the background, experimental design, key results, and the broader implications of this work, not just for gene regulation, but also for how we think about RNA's role in biology. This study focuses on the RFN element, which is a conserved RNA structure found in the 5' untranslated regions, also known as UTRs, of bacterial mRNAs involved in FMN biosynthesis and transport. FMN stands for flavin mononucleotide, an important coenzyme derived from riboflavin. What makes this RNA interesting is that it acts as an FMN-dependent riboswitch. Riboswitches are RNA elements that directly bind small molecules like FMN without the need for any protein factors. When FMN binds, the RNA changes its shape, which in turn regulates gene expression. The authors illustrate this clearly in Figure 1a, which shows the predicted secondary structure of the RFN element. It contains 6 stem loop regions that converge on a highly conserved junction. These conserved nucleotides are suspected to form the core FMN binding pocket. This figure gives us a structural context for the experiments that follow. The study aims to test whether the RFN element can bind FMN and regulate gene expression on its own, without proteins. To do this, they focus on 2 specific RNA leaders, the RibD leader, which controls genes for FMN biosynthesis, and the YPAA leader, which controls a riboflavin transporter. By working in vitro, they could isolate RNA function and determine whether RNA alone is responsible for FMN sensing. Flavin mononucleotide is a derivative of riboflavin, also known as vitamin B2. Inside cells, FMN acts as a key cofactor in many redox reactions, which are reactions that transfer electrons. It plays an essential role in cellular respiration and other metabolic pathways. Bacteria have specialized biosynthetic pathways for producing FMN, usually encoded in operons, or clusters of genes that are transcribed together. Because FMN is so important, cells need a way to regulate its levels precisely. If too little FMN is produced, essential metabolic reactions can't occur efficiently. But if too much is produced, the cells waste energy and resources. For this reason, bacteria have evolved feedback regulatory systems to control FMN synthesis and transport. One of these regulatory elements is the RFN element, which is conserved across many bacterial species. The RFN element is always found in the 5' UTR mRNAs involved in FMN pathways, suggesting that it plays a critical regulatory role. In Bacillus subtilis, the RFN element plays a central role in controlling genes involved in FMN metabolism. One key target is the RibDEHT operon, also called RibD, which encodes several enzymes for FMN biosynthesis. Normally, when FMN levels are high, the expression of this operon is downregulated, which prevents overproduction. Mutations in the RFN element eliminate this FMN-mediated repression, which tell us that the RFN structure is essential for sensing FMN or for transmitting the regulatory signal. Another gene under RFN control is YPAA, which encodes a riboflavin transporter. The mechanism is a bit different here. Instead of affecting transcription, FMN binding to the RFN element in the YPA leader is predicted to block the ribosome binding site, which prevents translation initiation. So, within the same organism, the RFN element appears to regulate gene expression through two distinct mechanisms, one at the level of transcription for RibD and one at the level of translation for YPAA. This dual functionality makes it an ideal system to study how RNA alone can exert regulatory control. Based on these observations, the authors hypothesized that the RFN element itself is an RNA aptamer. In other words, a structured RNA that can directly bind FMN with high specificity and affinity. Once bound, this interaction triggers a conformational change in the RNA that leads to regulatory effects. Specifically, they propose two mechanisms. First, for RibD, FMN binding induces a structure that causes premature transcription termination. And second, for YPAA, FMN binding sequesters the Shine-Dalgarno sequence, blocking translation initiation. To test this hypothesis, the authors took the regulatory regions of RibD and YPAA, cloned them, and transcribed them in vitro using T7 RNA polymerase. Then, they tested whether adding FMN to these RNA molecules could trigger structural changes and regulatory effects in the complete absence of proteins. This approach allowed them to isolate the RNA's behavior and determine whether it alone is responsible for FMN sensing. The experimental strategy involved several well-organized steps. First, they cloned the regulatory regions from the bacterial genome into plasmid vectors. This allowed them to produce clean DNA templates for RNA transcription. Second, they carried out in vitro transcription to synthesize radiolabeled RNA fragments corresponding to the RibD and YPAA leader sequences. Using the T7 promoter ensured strong, consistent RNA production. Third, they used inline probing, a biochemical technique that detects structural changes in RNA by taking advantage of spontaneous cleavage at flexible regions. This was critical for seeing whether FMN binding changes the RNA's structure. Fourth, they performed transcription termination assays to see whether FMN actually influences where transcription stops in the RibD leader. And finally, they used deoxyribozyme-mediated cleavage to map the precise transcription termination site at single nucleotide resolution. By combining all these methods, they built a strong case for the RFN element functioning as a riboswitch. This study relied on a variety of standard biochemical reagents and oligonucleotides. Synthetic DNA sequences for the regulatory regions of RibD and YPAA were purchased from the Yale Keck Biotechnology Resource Center. These oligonucleotides were purified using denaturing polyacrylamide gel electrophoresis and then recovered by eluting them from the gel in tris hydrochloride buffer with sodium chloride and EDTA, followed by an ethanol precipitation. For ligands, they use FMN, FAD, and riboflavin purchased from Cigna. These were added to the inline probing and transcription reactions to test specificity and binding strength. Radio-labeled nucleotides alpha-phosphorus 32 UTP and alpha-phosphorus 32 ATP were used to label RNA transcripts so they could be visualized on gels using a phosphorimator. Throughout the experiments, RNA and DNA fragments were analyzed using denaturing polyacrylamide gels, typically 6 to 10% acrylamide with urea to keep RNA unfolded. Gels were run for extended periods to get sharp, high-resolution bands. To generate transcription templates, the authors amplified the regulatory regions of RibD and YPAA from Bacillus subtilis genomic DNA. For RibD, they amplify nucleotides -61 to +302 relative to the transcription start site. For YPAA, they amplify -94 to +347. These PCR products were cloned into the PGM4Z plasmid using EcoR1 and BAMH1 restriction sites. The plasmids were then transformed into E. coli top 10 cells for propagation and verified by DNA sequencing. This cloning step was crucial because it created stable plasmid templates that could then be used for in vitro transcription. It also ensured that the exact regulatory regions were preserved for biochemical analysis without additional cellular factors interfering. Next, the authors used in vitro transcription to synthesize RNA fragments corresponding to the RibD and YPAA leaders. To do this efficiently, they replaced the native B subtilis promoter with the T7 RNA polymerase promoter using PCR primers. They also introduced a CC dinucleotide upstream of the transcription start site so that both transcripts began with GG. This is important because T7 RNA polymerase initiates transcription most efficiently with a GG start. The DNA templates were transcribed using either the standard T7 RNA polymerase method or the ProMega RiboMax kit, which is optimized for high-yield RNA synthesis. The RNA products were then purified on denaturing page and 5' end labeled with phosphorus 32 for detection. This gave them clean labeled RNA molecules that they could use for structural probing and transcription termination assays. By doing this step first, they ensured that all subsequent experiments used RNA molecules that were uniform, correctly transcribed, and free from any cellular contaminants. This level of control is essential when you're testing RNA structure and function outside of the cell. Essentially, they were building a simplified, purified version of the RNA system so that any behavior they observed could be confidently attributed to the RNA itself, not to the proteins or other factors. The inline probing experiments were designed to detect ligand-induced RNA folding changes. This method relies on the natural tendency of flexible RNA regions to cleave themselves over time in the presence of magnesium. Structured regions are protected, while unstructured regions are more prone to cleavage. They also incubated radio-labeled RNA at low concentrations in a buffer containing trihydrochloride, magnesium chloride, and potassium chloride for about 40 hours at room temperature, with and without the ligands FMN, FAD, or riboflavin. Reactions were protected from light to avoid FMN degradation. After incubation, the RNA was run on long polyacrylamide gels. By comparing cleavage patterns in the presence and absence of FMN, they identified which regions of the RNA changed structure upon binding. This data also allowed them to estimate apparent KD values, referring to the equilibrium dissociation constant, which measures the affinity of FMN binding by plotting the fraction of cleavage at specific sites versus ligand concentration. In other words, they could actually see how tightly the RNA binds FMN and which parts of the molecule move or stabilize during binding. This approach gives a visual biochemical fingerprint of how RNA structure responds to ligand interaction. It's an elegant way to show that RNA itself is not static. It's dynamic and capable of adopting new conformations based on small molecule signals, much like a protein receptor would. To test whether FMN binding affects transcription, they performed transcription termination assays using T7 RNA polymerase. They transcribed DNA templates containing the RibD leader sequence and reactions containing radio-labeled UTP. FMN, FAD, or riboflavin was added to some reactions at 100 micromolar. The reactions were then incubated at 37 degrees Celsius for 2 hours and then analyzed on denaturing gels. By quantifying the ratio of full-length to prematurely terminated transcripts they could determine whether FMN caused increased transcription termination. They also made mutant templates where they changed the length of the U-rich terminator track to test how important it was for termination. Templates with fewer than 6 U residues lost FMN-dependent termination, confirming that this U-rich region is essential for the mechanism. Finally, they mapped the exact termination site with the RibD leader using a deoxyribozyme cleavage strategy. Both full-length and FMN-terminated transcripts were generated. Then a 10 to 23 deoxyribozyme was used to cleave the RNA after nucleotide A222. This created two fragments, FL for full-length and T for terminated transcripts. These fragments were 5' labeled and subjected to partial RNase T1 and alkali digestion to generate size markers. When run on a 10% denaturing page, this allowed them to determine the precise location of the termination site by comparing band migration. They found that termination occurs between nucleotides U261 and U263 in the U-rich tract downstream of the terminator stem. This provided direct single nucleotide resolution evidence of FM-dependent termination in vitro. What's really important here is that this experiment tied the biochemical data back to a functional output, whether the RNA actually turns transcription on or off. It's the step that connects structure to function and shows that FMN binding has a real regulatory consequence. To summarize the methods, the authors cloned regulatory sequences for RibD and YPAA. They transcribed them in vitro with T7 polymerase and radio-labeled the RNA. They then used inline probing to map structural changes upon FMN binding, transcription termination assays to test regulatory effects, and deoxyribozyme mapping to pinpoint where termination happens. By following this logical experimental sequence, they were able to isolate RNA function from cellular context and prove that the RFN element can act as an FMN riboswitch. These methods combine classic molecular cloning and transcription with clever RNA biochemistry, and together they provide very strong evidence for the central hypothesis of the paper. Excuse me. It's also worth noting that these methods become foundational for later riboswitch studies. Many of the same techniques like inline probing and termination assays are still used today. This study essentially set the standard for how researchers test whether an RNA sequence can function as a metabolite sensing switch. The first major result is shown in Figure 1, which examines whether the RFN element can bind FMN directly without proteins. The author used a 165 nucleotide RNA fragment from the RibD leader and analyzed its structure using inline probing, which detects spontaneous cleavage at flexible regions of the RNA. In Figure 1b, they illustrate this probing strategy. In the absence of FMN, the RNA shows a baseline cleavage pattern. But in Figure 1c, when FMN is added, certain conserved nucleotides at the multi-stem junction becomes more protected from cleavage, while other regions show increased cleavage. This indicates that the RNA undergoes a specific structural rearrangement upon FMN binding. Because these experiments were done in a purified RNA system with no proteins, the data demonstrates that the RFN RNA alone can bind FMN and form a stable FMN-bound conformation. This established the RFN element as a true RNA aptamer. This was a big deal at the time because it showed that RNA could function as a receptor for a small molecule, something that was previously thought to be a job only for proteins. It also supported the broader concept that RNA structure is sophisticated enough to recognize complex metabolites with high specificity. Figure 2 addresses whether the RibD RFN element binds FMN specifically or if it also interacts with related flavins like riboflavin or FAD. In Figure 2a, the author shows inline probing titrations using FMN at increasing concentrations. You can see that the cleavage pattern changes at low nanomolar levels, indicating tight binding. They They calculated an apparent KD of about 5 nanomolars for FMN, which is exceptionally strong for RNA small molecule interactions. In contrast, Figure 2b shows similar titrations using riboflavin and FAD. Riboflavin, which lacks the phosphate group, only affects the RNA at micromolar concentrations, around 3 micromolars. The FAD, though, shows intermediate binding at around 300 nanomolars. These results show that RFN RNA strongly discriminates in favor of FMN, particularly recognizing the phosphate component. This level of molecular discrimination is similar to what is seen in protein-based riboswitches. So not only does this RNA bind FMN tightly, it's highly selective. That means that the RNA can distinguish between molecules that differ by just one chemical group. This gives us a glimpse into how evolution shaped these RNAs to respond only to the right metabolite, avoiding false activation by similar compounds. The functional consequence of FMN binding is shown in Figure 3. Here, the authors use the 304 nucleotide Rib-D RNA that includes the terminator region. They performed in vitro transcription assays with T7 RNA polymerase in the presence or absence of FMN. Figure 3b shows the transcription product separated on a gel. In the absence of FMN, about 10% of the transcripts terminate early, giving a shorter product. But when FMN is added, this increases to around 30%. This is a clear FMN-dependent increase in premature termination. The model is shown schematically in Figure 3a. FMN binding stabilizing a structure in the leader RNA that forms a terminator hairpin, followed by a U-rich tract. This hairpin causes the polymerase to dissociate, stopping transcription before the downstream genes are transcribed. These results demonstrate that the RFN element doesn't just bind FMN, it uses this interaction to regulate transcription termination, behaving like a riboswitch. This is the functional proof point of the whole study. It shows regulation in action, and it also illustrates a principle seen in many bacterial riboswitches. When the metabolite is abundant, transcription of biosynthetic genes stop, conserving energy and resources. That's an elegant example of feedback regulation at the RNA level. To pinpoint exactly where FMN-induced termination occurs, the authors used the clever mapping approach shown in Figure 4. They generated full-length and terminated transcripts, then used the deoxyribozyme to cut after nucleotide A222. The resulting 3' fragments were labeled and run alongside partial RNase T1 and alkaline digest to create size markers. As shown in 4b, the termination site mapped precisely to nucleotides U261 through U263 in a U-rich stream downstream of the terminator hairpin. They then created U-track mutants to test the importance of this region. In Figure 4c and 4d, you can see that when the U-track was shortened to fewer than 6 uridines, FMN-dependent termination was essentially lost. This confirms that the U-stretch is critical for the termination mechanism, just like in classical row independent transcription terminators. Combined with Figure 3, this nails down how FMN binding leads to transcriptional regulation in Rib-D. This figure adds a layer of mechanistic precision. It's not just showing that termination happens, it tells us exactly where and why. Mapping to the nucleotide level makes the argument extremely strong and shows that the RNA-based regulation can be dissected with the same precision as protein mechanisms. The last figure, Figure 5, shows that not all FMN riboswitches use the same regulatory strategy. The YPA RNA, which is 349 nucleotides long, doesn't seem to terminate transcription. Instead, FMN binding affects the Shine-Dalgarno sequence, which is the ribosome binding site. Using inline probing, the authors found that in the absence of FMN, the SD sequence region is flexible and exposed, meaning ribozymes can bind and initiate translation. But in the presence of FMN, the RNA folds differently, sequestering the SD sequence into a stem loop structure. This is depicted schematically in Figure 5. Excuse me. This folding prevents the ribosome from accessing the SD sequence, effectively switching translation off. So while the RibD RNA uses transcription termination to control gene expression, the YPAA RNA uses translation initiation control through SD sequestration. This highlights the versatility of FMN riboswitches in regulating gene expression. This diversity in mechanism really underscores how adaptable RNA can be. The same metabolite FMN can trigger entirely different outcomes depending on the RNA context. It's like one signal molecule speaking two different languages of regulation. To summarize the results in the figures, Figure 1 established that the RibD RFN RNA binds FMN directly and changes structure without proteins. Figure 2 showed that this binding is highly specific for FMN with nanomolar affinity and discriminates against related flavins. Figure 3 demonstrated that FMN binding leads to transcription termination in vitro. Figure 4 mapped the termination to a precise U-rich stretch and confirmed its necessity through mutagenesis. Finally, Figure 5 revealed that the YPAA uses a different mechanism, folding to hide its Shine-Dalgarno sequence upon FMN binding, thereby blocking translation initiation. Together, these figures provide a complete mechanistic picture. FMN riboswitches use direct metabolite binding to control gene expression through both transcriptional and translational mechanisms. This logical flow from structure to specificity to function to precise mechanism makes this one of the most convincing early riboswitch papers. It ties every observation back to the core idea that RNA alone can sense and respond to metabolites, something that reshaped molecular biology thinking in the early 2000s. The authors conclude that the RFN element is a natural RNA aptamer, essentially an RNA receptor for FMN. Across their experiments, they showed that RFN RNA binds FMN with very high affinity in the low nanomolar range and with strong specificity compared to related molecules. Functionally, this aptamer is integrated into two distinct riboswitch mechanisms in Bacillus subtilis. As demonstrated in figures 3 and 4, FMN binding in the RibD leader triggers premature transcription termination, which is an inefficient way to shut down expression of large biosynthetic operons when FMN levels are sufficient. In contrast, as shown in figure 5, FMN binding in the YPAA leader causes structural sequestration of the Shine-Dalgarno sequence, thereby blocking ribosome binding and translation initiation. This mechanism provides a rapid regulatory response at the level of translation. Together, these findings establish that RNA alone, not proteins, can directly sense a metabolite and regulate gene expression in two different ways. This dual functionality really shows evolutionary efficiency. One metabolite, two regulatory outcomes, all mediated by RNA, is a powerful example of how small structural RNAs can control big decisions in gene expression. These discoveries have broad implications for how we think about gene regulation. The ability of RNA to bind small molecules and directly regulate gene expression supports the RNA world hypothesis, which proposes that early life relied heavily on RNA for both catalysis and regulation before proteins evolved. The RFNN riboswitch is a clear modern example of RNA performing both sensing and regulatory roles. Given that FMN is just one metabolite, the authors suggests that many more riboswitches likely exist in bacteria, controlling a variety of pathways and using diverse regulatory strategies. Some may control transcription, others translation, and potentially other steps as well. Overall, this work highlights RNA as a central regulatory molecule, not just a passive messenger. It uses structure, folding, and binding specificity to control gene expression efficiently. This study also inspired a wave of research that uncovered dozens of of additional riboswitch classes. It shifted the perspective from seeing RNA as just an information carrier to seeing it as an active, intelligent molecule capable of responding to the cell's metabolic state in real time. To wrap up this presentation, the study by Winkler and colleagues provided the first compelling evidence that RNA structures, specifically the RFN element, can act as a fully functional riboswitch by directly sensing metabolites like FMN. They showed that RNA alone can bind FMN with nanomolar affinity and use this interaction to control gene expression in two different ways: by triggering transcription termination in the RibD operon and by blocking translation initiation in the YPAA gene. This dual mechanism not only highlights the versatility of RNA base regulation, but also reinforces the concept that RNA is more than just an intermediary between DNA and proteins. It can sense, respond, and regulate. These findings also support the RNA world hypothesis by showing that RNA still retains ancient protein-independent regulatory capabilities in modern cells. And perhaps most excitingly, it opens the door to discovering many more riboswitches that control gene expression across diverse bacterial systems. So ultimately, this paper didn't just characterize a single RNA element, it helped reshape how we think about RNA's role in biology. It bridged the gap between ancient RNA functions and modern genetic control, giving us a glimpse of how life might have regulated itself before proteins took over. It's one of those rare papers that changes both experimental technique and theoretical perspective.